Hello, good morning, everyone. My name is Delenasaw Yewhalaw from Tropical and Infectious Disease Research Center of Jimma University, Ethiopia. And my talk will be on Anopheles stephensi distribution, bionomics, and efficacy of candidate larvicides for the control of stephensi larvae in Ethiopia. Here is an overview of malaria in Ethiopia. Malaria is a major public health and socio-economic problem in Ethiopia. Generally, there is an upsurge of malaria in Ethiopia since 2020, and over 75.2 million people live in areas at risk of malaria infection since 2023. Malaria is seasonal and unstable in most areas of the country. Major transmission occurs from September to December, following the long rain, and minor transmission season occurs from April to May, following the short rain. Malaria mainly occurs at an altitude less than 2,000 meters above sea level in Ethiopia. It historically is characterized by cyclic epidemics. Here, this map shows the malaria risk stratification of Ethiopia based on environmental and climatic risk factors, and also annual parasite incidence. In general, the high malaria risk infection and larger number of cases are confined in areas bordering Eritrea, Sudan, and South Sudan, and also in eastern and northeastern parts of Ethiopia. Coming to the malaria vectors in Ethiopia, the native primary vector of malaria in Ethiopia is Anopheles arabiensis, which has a wide distribution all over the country and which accounts for 99.5% of all cases. And there are also native secondary malaria vectors, which have limited distribution in the country and minor role in transmission, and which include Anopheles funustus, Anopheles pharonsis, and Nili. Anopheles kaustani was a suspected vector, but it recently has been incriminated as a malaria vector in Ethiopia, and it has also a wide distribution. And finally, an invasive vector, Anopheles stephensi, has been detected in Ethiopia in 2016, and now it's spreading in most areas of the country. Plasmodium falciparum and Plasmodium vivax are the two important malaria parasite species co-occurring in Ethiopia, and their prevalence varies according to locality and season. This shows the global native territories and recent spread of Anopheles stephensi. Anopheles stephensi is an efficient vector, is known to be efficient vector in Southeast Asia and the Mediterranean region. And however, it has been recently detected in Africa, especially in 2012 in Djibouti, and then in 2016 in Ethiopia. And now it is extending its geographic range to West Africa. Anopheles stephensi is not a single species, rather it has biological forms. 3 biological forms are there, and this includes type form, intermediate form, and mysorensis form. The type form is more of anthropophilic, and it is an efficient malaria vector in urban areas, whereas the intermediate form is still anthropophilic, and it is a malaria vector in rural areas. And these two forms occur in sympatry in most breeding sites. The last one, the third one, the mysorensis form, is more of zoophilic and is less important in terms of malaria transmission. This shows the current distribution and suitability map of Anopheles stephensi in Ethiopia. Actually, as I have mentioned, Anopheles stephensi has been detected in 2016 in the eastern part of Ethiopia for the first time, but it has been detected later in northeastern and also northwestern and southwest and southern Ethiopia. Right now, there are about 53 sites which are positive for Anopheles stephensi in Ethiopia. Anopheles stephensi breeds in both artificial and natural breeding habitats in both urban and rural areas. In Ethiopia, Anopheles stephensi breeds in many different types of artificial breeding habitats or in a range of artificial water containers, which are mainly used to store water for brick making, and it also breeds in natural breeding habitats, which include in marsh areas, stream beds, and also in river edges. This also shows the distribution of immature Anopheles stephensi in natural and artificial breeding sites in Ethiopia. In some sites are positive for immature stages. Some artificial breeding sites, some sites are positive. Some artificial breeding sites are positive for Anopheles stephensi immatures, and in other sites, some natural breeding sites are also positive for Anopheles stephensi immature stages, and in some sites, both artificial and natural breeding habitats are positive for Anopheles stephensi immature stages. This shows the larval density and mosquito species composition by season, habitat type, and setting. Actually, larvae of Anopheles, Anopheline mosquitoes are available during the dry and rainy season. Here we can see higher larval density has been documented during the rainy season as compared to the dry season, but larval mosquito larvae are also common during the dry season. This includes also larvae of Anopheles stephensi. So when you look at the proportion of Anopheles stephensi in artificial and natural breeding habitats in urban setting, and also in artificial and natural breeding sites in peri-urban setting, and also artificial and natural breeding sites in rural setting, we can see that the proportion of Anopheles stephensi was higher in artificial breeding sites in urban areas as compared to the proportion of Anopheles arabiensis and other species. And in natural breeding habitats in urban setting, actually the proportion of Anopheles arabiensis and other species was higher than the proportion of Anopheles stephensi. So a similar trend was observed in artificial and natural breeding habitats in peri-urban areas. And however, in rural areas, higher proportion of Anopheles stephensi was recorded as compared to Anopheles arabiensis and other species. But in rural areas, especially in the natural breeding sites, no Anopheles stephensi was documented or recorded. We have been conducting a study to evaluate the efficiency of trapping methods for adult Anopheles stephensi collection in Ethiopia. So as you see, when you look at the trapping efficacy of Procopac aspirator as compared to CDC light trap, Procopac aspirator catches higher proportion of Anopheles stephensi than CDC light trap. This also shows Anopheles mosquito density by trapping method. And we have evaluated the efficacy again of different trapping methods in different sites in terms of Anopheles stephensi patches. So BG Pro trap and Procopac aspirator traps or catches more Anopheles stephensi or higher density of Anopheles stephensi than Anopheles arabiensis in both sites. This shows the outdoor and indoor Anopheles stephensi collection by site and trapping method in Ethiopia. Here, we have been collecting Anopheles stephensi both indoor outdoor using CDC light trap and Procopac. However, the catches, Anopheles stephensi catches was small, so small, so it was difficult really to say that the proportion of Anopheles stephensi from outdoor was higher than indoor, or vice versa, because due to the low number of catches, it was difficult to see the difference in outdoor and indoor collection in terms of Anopheles stephensi catch in the study. This shows the blood meal source of Anopheles stephensi in Ethiopia. So Anopheles stephensi feeds on a range of vertebrate hosts from human to chicken. However, Anopheles stephensi prefers to feed on animals, especially on ovines or goats, to humans. This shows actually parasite infection in Anopheles stephensi or plasmodium infection in Anopheles stephensi. So Anopheles stephensi collected from different sites were found to be positive for plasmodium infection. And this shows that Anopheles stephensi has a potential to transmit malaria in Ethiopia. We have also conducted insecticide susceptibility tests on Anopheles stephensi against different insecticides, and the results showed that Anopheles stephensi developed resistance to pyrethroids, organophosphates, and even to new candidate insecticide class like neonicotinoids, that is against clothianidin. So it's difficult to think of using chemical insecticides for the control of Anopheles stephensi in Ethiopia. We have been conducting to assess or evaluate the residual efficacy of some novel candidate larvicides, which include Sumilar 2MR and Sumilar 0.5G, and also a conventional larvicide, Abati-1 SG, against Anopheles stephensi larvae in Ethiopia. And we found that Sumilar 0.5G lasts for from 3 to 4 weeks, and the conventional larvicide Abati-1 SG lasts for 5 to 6 weeks, while the Sumilar 2MR lasts for 9 months, which means the percentage adult Anopheles stephensi emergence inhibition lasts for 9 months for Sumilar 2MR. This shows the control of Anopheles stephensi larvae using Sumilar 2MR in Ethiopia. We have applied the same concentration of similar to MR in different volumes of water, like in 100 liter, 250 liter volume of water, and 500 volume of 500 liter of water. And we found that in both volumes of water, the percentage emergency inhibition of Anopheles stephensi lasts for 31 weeks. Therefore, similar to MR is a potential candidate larvicide to control or for the control of Anopheles stephensi larvae in Ethiopia. In conclusion, Anopheles stephensi breeds both in urban, peri-urban, and aquatic environments. It breeds both in artificial and natural habitats. Larval density shows seasonal variation, and this invasive species can transmit Plasmodium parasites and potentially transmit malaria in dry season. Anopheles stephensi prefers to feed on animals to humans. It rests both indoor and outdoor, and stephensi is resistant to most public health insecticides. Similar to MR, was found to be a potential larvicide for the control of Anopheles stephensi larvae. Here are the knowledge gaps. That is, the seasonal dynamics of stephensi in Ethiopia is not well understood. Most prolific breeding habitats of Anopheles stephensi still are not well understood. The role of Anopheles stephensi in malaria transmission as compared to native malaria vectors is not well documented. Optimal surveillance tool for sampling Anopheles stephensi are lacking, and the unique cryptic resting sites of adult Anopheles stephensi is not well known. Anopheles stephensi have the potential of replacing native vector species over time is not well understood, and optimal intervention strategies to control Anopheles stephensi is not well understood, and the form of the species, which is important in transmitting malaria in Ethiopia, is not yet understood. Finally, I would like to thank our collaborators, the ICMR Ethiopia project collaborators, and the SEALS Ethiopia collaborators. I would like also to thank the RAFT group for inviting me to present our work in the MIM conference. And thank you all for your attention. Thank you very much.